This method can help answer key questions in the field of ophthalmology and retinal imaging, such as imaging and quantification of blood-retinal barrier disruption and retinal capillary functions.
The main advantage of this technique is that it can obtain a large field of view, three-dimensional, multi-contrast retinal imaging by using an oblique scanning laser in one single raster scan.
The implications of this technique extend toward diagnosis of diabetic retinopathy and other retinal diseases, because oSLO can obtain high-contrast images of retinal microvasculature down to single capillaries in 3D.
Though this method can provide insight into retinal imaging, it can also be applied to other imaging systems using conventional objective lens, such as in vivo imaging of mouse cortex.
A supercontinuum laser source is used as the system laser source for the oblique scanning laser ophthalmoscopy or oSLO setup. The visible light range is separated from higher wavelength range by the first dichroic mirror. The light spectrum is expanded with a pair of dispersive prisms after the beam passes through a polarization beam splitter. A slit is used to select the excitation wavelength range. And a reflective mirror reflects the filtered beam back to the prism pair to couple the light into a single mode fiber.